Hello, everyone, and thank you for joining today's live rebroadcast of an ASHG Digital CoLab webinar we hosted earlier this year. We thank ASHG for the initial opportunity, and we're excited to share this today to make the content available beyond the ASHG community. This webinar will address how leveraging population-scale PacBio long-read sequencing can help to uncover structural variation driving disease associations with insights from the All of Us initiative. Before I pass it over to our speakers, I just wanted to quickly mention some housekeeping items. First, we will be fielding Q&A questions. If you have any questions, please submit in the questions module in your attendee control panel, and PacBio or one of the speakers will get back to you. We'll also have some related PacBio literature available in the handouts module. After the webinar, this recording will be available on demand on our website and YouTube channel, and we'd be very grateful if you could take a moment after the talk to fill out our post-webinar survey. With that, I'm going to hand things over to Aaron Wenger, who is a Senior Director of Product Marketing at PacBio. He'll give us an overview of PacBio technology as well as some exciting product updates, and then we'll hand it over to our main speaker, Professor Michael Schatz.
Great. Thanks, Nina. As Nina said, I'll focus on some of the tools that PacBio has developed for large-scale structure variation studies. And first among these is the core sequencing technology, so the PacBio HiFi sequencing chemistry. Fundamentally, this technology delivers a superior, more complete class of genome. The core capabilities of the sequencing chemistry are that it generates long reads up to 25,000 base pairs in length with extremely high accuracy, uniform coverage of the genome, and it's sequencing single native molecules and thus able to see not only the sequence of those molecules, but also epigenetic modifications like 5-methylcytosine. When applied to whole genome sequencing, this gives a view of all classes of variation in the genome. So while with short read sequencing, you'll get a view of primarily single nucleotide variants and small indels, with HiFi whole genome sequencing, you see those single nucleotide variants and small indels, but also larger structural variants, which are typically defined as genetic differences that involve 50 or more base pairs. You'll see variation in duplicated regions of the genome, in tandem repeats, and you see this variation not just in isolation, but also phased into maternal and paternal haplotypes directly based on the read data and linked to DNA methylation. One of the key applications of this sequencing technology in human genetics has been the study of rare disease. And I think a nice illustration of the power of a comprehensive genome in understanding rare disease was from this study last year, or published last year from Children's Mercy Kansas City, where they applied HiFi long-read whole genome sequencing, which is shown in orange in the plot here, and a number of standard of care methods to look at a few hundred individuals with a putative rare disease. And they were able to identify a genetic cause for the disease in a higher percentage of individuals using the more comprehensive genome. So here it's 37% compared to 27% for the standard of care. And importantly, they were able to do so faster because rather than doing serial testing with a bunch of different modalities, they had a single comprehensive genome with the HiFi whole genome sequencing. The HiFi sequencing is available on two different sequencers today. The first is shown on the left here, the Vega system. This is a system designed around accessibility. It's a benchtop system with very simple user experience. It's designed to be in labs that focused on genome sequencing, but also targeted in RNA sequencing. And this has been a good entry point for a lot of labs into using HiFi sequencing. So more than 60% of Vega owners are first-time PacBio customers. On the right is the second system where you can access HiFi sequencing technology, which is the Revio system. This is our production system capable of 2,500 genomes per year. And this is designed to support large projects in carrier screening, population genetics, and clinical research. And it's the platform that was used by Children's Mercy Kansas City that I cited on the previous slide, and also being used in larger projects like All of Us. Beyond the sequencers themselves, PacBio has also developed a number of different computational methods to allow insight to come from the HiFi sequencing data. These span from read alignment through different variant calling approaches. The tools are available as individual modules for those looking to do just a particular study of the genome. And they're also integrated together in what we call the WGS variant pipeline. It's kind of a single integrated workflow that takes in HiFi reads for a genome and applies all these different methods and gives a unified call set on the out. There are two specific methods that I wanted to go into a little more detail today that are relevant to today's topic. The first of those is Sawfish, which is PacBio's unified structural variant and copy number variant caller. This is a method that takes reads aligned to a genome. It detects candidate breakpoints or candidate structural variants from alignment abnormalities. And then at each of these regions where a candidate is identified, it will perform an assembly of the reads that map to that region. This produces then the two haplotypes that are present at the locus. Those two haplotypes are mapped back to the reference genome to call the variant and assign a zygosity. The method also integrates depth-based information, which allows it to say copy number variants that are longer than that 10 to 15,000 base pairs that you'd get from a typical assembly-based approach. Sawfish is primarily used to look at individual human samples, but it also provides a joint calling capability that supports up to 10 samples. So if you're looking at a family or a small cohort, Sawfish provides a way to get a unified single VCF output that has genotypes assigned to every structural variant seen in any individual in the cohort. For larger sets, beyond this scale of 10, when you're looking at hundreds, thousands, or as we'll hear from Mike today, thousands and tens of thousands, we've developed a method called SVX that merges variant call sets from large cohorts. So here, the approach for SVX is that each individual in the cohort has variant calling applied separately to them. So if you're, say, looking at 1,000 individuals, it'd be running Sawfish 1,000 times. Producing 1,000 VCFs, and then SVX would take those 1,000 VCFs and unify them, producing a joint call set. To give an example of its application, show here on the right the application of SVX to a 28-person pedigree. Across these 28 individuals, from the individual call sets, there were 952,790 structural variants that were called. And when these are merged with SVX, it consolidates down into 92,367 merged variants. So most of the variants are polymorphic across this cohort and thus are merged into a smaller number of unified calls. And then this histogram shows for insertions and deletions the frequency distribution seen in this relatively small, in this case, cohort. So you can see on the left, a number of variants are singleton, seen in only one individual, but there are also a number of variants that are seen in multiple individuals in the cohort and some that are present in all individuals of the cohort. This approach of gathering a bunch of genomes, individually calling structural variants, and then merging them into a joint call set was applied by the Consortium for Long-Read Sequencing, which is an effort to produce what's called the CoLoRSdb. It's an allele frequency database for HiFi-based structural variant calls. In this case, it uses 1,400 human genomes, and the variant calls from this set are available at colorsdb.org. This frequency database provides a really great control when trying to interpret structural variants called from long-read sequencing, from HiFi sequencing. And on the bottom, I'd show an example of how important it is to have a long-read matched control when looking at structural variants from long-read sequencing. So here we have an individual healthy female where HiFi sequencing plus analysis with Sawfish will identify 28,161 structural variants. If you then ask how many of those are unique when you compare to different frequency databases, you can see compared to the CADD-SV short read based structure variant database that the vast majority of the variants in this individual are not seen in the control database, so 23 of the 28,000. While comparing to CoLoRSdb, you see that only 6,000 of the 28,000 variants appear to be unique to that individual. That just shows the importance of having a comprehensive control set to go with your comprehensive set in the individual. So So Sawfish is really focused on these kind of classically defined structural variants, deletions, insertions, translocations. We also have a method called target or TRGT that specifically looks at tandem repeat structural variation, where you have individual motifs where copies are either added or reduced in particular alleles. And Target has an explicit model for millions of tandem repeats across the human genome. It knows where they occur, what motifs comprise them, and what the basic structure of motifs in the repeat are. And it's able to give a very clear interpretation of these difficult regions. Just like the structural variant calling plus merging approach was used to produce the CoLoRSdb frequency database, Target was applied to a number of human genomes to produce the TRExplorer frequency catalog. This is available in this really nice interface at trexplorer.broadinstitute.org. And it has a catalog of 4.8 million tandem repeats across the human genome. And this is showing just a region of this catalog around the gene FMR1, where there's a known pathogenic tandem repeat, with the one shown in red here. And you can see the type of information you get in this catalog. So it shows you the coordinates in the HU38 reference genome, the motif that comprises the repeats, and then on that second column from the right, the allele distribution across 256 humans. So you see how many people have 8 copies of the repeat, 20 copies of the repeat, and so on. And this is really useful for understanding in an individual case how unusual a particular repeat copy may be. So beyond developing this kind of core sequencing chemistry and then the sequencers and the analysis tools for looking at the data, PacBio is also continuing to push on the economic feasibility of using the platform. And most recently, we announced what's called the SPRQ-Nx chemistry. This is a new chemistry for the Revio production scale system that enables multi-use of the SMRT Cell sequencing consumable. And the ability to use this consumable multiple times dramatically drives down the cost of generating a human genome. With this SPRQ-Nx chemistry, a HiFi human genome costs around $300 to $350, which is around a 30 to 40% reduction from the current chemistry, and a 10 times reduction from where it was only a few years ago in 2022. This chemistry is in early access now, been in early access since the end of last year, and it is planned for full commercial release in the middle of this year. We're really excited to see how the improvement in the output you get from the system and the cost will allow even larger studies to happen with HiFi sequencing. And with that, it's a good opportunity to introduce our main speaker for today, who will be talking about his work on a really large-scale study using HiFi sequencing. And that's Professor Michael Schatz, who's the Bloomberg Distinguished Professor of Computer Science and Biology at Johns Hopkins University. Mike's a leader in computational genomics, genome assembly, and large-scale variant analysis for human and non-human genomes. He's developed a number of really groundbreaking and critical methods for looking at long-read structural variants. He's been a key contributor to national genomics infrastructure in the United States, including the ANVIL program and the All of Us research program. And he played a central role in the telomere to telomere consortium that delivered the first complete human genome, for which he was recognized as a member of the Time 100 Most Important People in the World list in 2022. And he's also received a number of academic honors, including a Sloan Fellowship and an NSF Career Award. He's always a great speaker, and I'm excited to see what he has to show us today. So with that, Michael, I'll turn it over to you.
All right, Aaron, thank you so much for that introduction. And thanks to all of you for joining today. Let me go ahead and share my screen. All right, hopefully everybody can see that right now. So thank you very much for that introduction. So as Aaron mentioned, I'm a professor at Johns Hopkins, although today I'm really here on behalf of the All of Us Long Read Working Group to present some of our latest findings using the HiFi technology specifically. So the short title for this talk today is Structural Variations Are Major Drivers of Traits in Disease. And this has been kind of really a long-going effort, an interest of mine for many years now to really kind of dig into the foundation of all these different traits and different diseases here. So I asked ChatGPT and DALL-E to make kind of a cutesy little image here. So this is a problem I've been thinking about for many years now. Today I'm focusing primarily on human genetics. I will say I've also spent a lot of time thinking about animal genetics and plant genetics and beyond. And I really will say that HiFi technology in particular has really unlocked a huge amount of the genome that has previously been unexplored. And it's just been really, really exciting to kind of dive into this. So today I'm going to be talking primarily about structural variations, although as you heard from Aaron, things like tandem repeats and sort of other parts of the genome are also very interesting. But today I'm really focused on structural variation. So in the literature, in the field here, these are any types of variants at least 50 bases in size. And that's sort of just a working definition. Sometimes we focus on larger, sometimes we focus on smaller, but in the literature here, it's really 50 bases. So this comes in many different forms. So these can be insertions, these can be deletions, these can be duplications, inversions, and combinations thereof where we get these sort of complex variation types. I should comment, this is not a new idea. So this is a really nice review article from almost 15 years ago talking about these types of structural variations and how you might find them using a variety of different methods, looking at how the reads align, how they assemble, looking at the depth of coverage, if you get sort of split read sort of mapping. These have been studied for a long time. So there's a lot known about them, but also there's new opportunities thanks to the new technology. So in a typical healthy person, we're going to have on the order of, say, 25,000 structural variations. The majority of these will be insertions and deletions. I should say the vast majority of these will be insertions and deletions, plus on the order of, say, a few hundred duplications, maybe a few tens of inversions that are present there. So the vast majority of these are these insertions and deletions. Now, if you reflect on this, you might be, okay, that's exciting. But a typical healthy human genome has on the order of a few million SNPs and a few 100,000 indels. You might think, oh, there's many fewer of these structural variations, so maybe they're not so important. But it's the complete opposite, right? Because these structural variations are much larger by definition. If you kind of look at a typical person, most of the variation from one person to the next is actually in structural variation. Even in a healthy sort of germline genome, we're going to have more than 10 megabases of variation captured through structural variation, whereas you'll have a few million bases in SNPs and maybe a million bases or so in small indels. So the majority of variation from person to person is actually in structural variation. Shown here on the right, this actually comes from all of us study. This is at a population scale. You get sort of this large number of smaller structural variations on sort of the 50 to 100 base regime. And then this long tail of variation out to kilobases, tens of kilobases and beyond, where you can have a few of those per person, and then larger numbers of those in a population there. So structural variations, yes, they're fewer, but they have in total they actually contribute more variation than any other class of variation. And because they're larger, they can have a really profound impact on, say, gene structure, gene regulation, where you can knock out whole genes, you can knock out entire regulatory elements through a single event, whereas, say, a SNP may have a more modest impact on the variation that is present there. Now, they've been known about for a long time, but the field has really changed in the last few years, right? In a couple years ago, Nature Methods declared long-read sequencing, the method of the year. We wrote a review article sort of capturing, we're really entering this era where we can have complete genomes, complete transcriptomes, complete epiomes, thanks to this really accurate long-read sequencing. And I'd say really, in terms of accuracy, HiFi sequencing today is the indisputed champion. You just get sort of excellent quality from the data. Average quality is, well over 99%. Many of the reads are perfect, which if you're looking for variation, if you're looking for assembly, having long perfect reads doesn't get any better than that. So it's really an exciting time to be working with HiFi sequencing. As Aaron pointed out, HiFi was really the star of the show for that first telomere to telomere reference genome. In red here are all the parts of all the different human chromosomes that we were able to fill in for the first time, primarily using HiFi sequencing. So every chromosome was improved through this. And things like, centromeric sequences, acrocentric sequences, subtelomeric regions, segmental duplications, and beyond were massively improved through the use of long-read sequencing, and particularly HiFi sequencing. And then there's a whole ensemble of papers commenting on how this new reference genome and, thanks again to long-read sequencing, how we're able to improve our study of segmental duplications, understand, say, centromeres, repeat elements, epigenetic patterns, and then our team kind of really led the study of how this new reference genome and long-read sequencing improves our analysis of human genetic variation. So it's really been sort of a comprehensive improvement across the board in sort of any sort of genetic and genomic analysis. It gets improvement through long-read sequencing. Now, I should say that first telomere genome, that was really exciting as a reference, but a real passion of mine is how can we use this to improve clinical interpretation as well? I should comment, this is not a new idea. Going back almost a decade now, we've been thinking about how long-read sequencing, and in particular PacBio sequencing, could improve, say, cancer genomics. Back in 2018, we sequenced a breast cancer cell line called SKBR3, and we discovered, many, many thousands of structural variations that had never been seen before, including things like gene fusions, all kinds of complex rearrangements that were previously uncharacterized. In 2020, we moved this into clinical settings where we're able to look at patient samples for the first time. And in a breast cancer patient, for example, we're able to find a repeat expansion, a structural variation in BRCA1. This is the most important risk factor gene in breast cancers. And this variant was completely undetectable using short-read sequencing. So we're really excited about some of these possibilities. And for many years now, kind of anecdotally, we could see, an example of this, an example of that. But we just didn't have yet the opportunity to look at this in a very comprehensive way, to look over many, many people, to look in a very comprehensive way. Well, what is the role of structural variation? What is the role of these more complex variation at a population scale? But I'm very excited to say, thanks to the All of Us initiative in the United States, we're able to do this at a population scale, at a biobank setting for the very first time. So this has been a dream of mine to move beyond these sort of individual cases and to be able to do this systematic study looking at truly population scale using HiFi sequencing. So I'm guessing on the call here today, many people are familiar of All of Us. Right, this is a major initiative in the United States. It's arguably one of the largest initiatives. The long-range goal here is to collect genomic resources as well as electronic health records and other personalized data for 1 million US citizens. And the goal here is really to have a biobank and a platform to understand human health very, very comprehensively. So at this scale, we can really dissect the genetic bases of many common diseases and also rare diseases. Really understand sort of treatments that work for different people and backgrounds, and really just sort of hopefully make progress on sort of shaping the future of human research. This is a major initiative that's been underway for many years now. In terms of the biobanking, more than 500,000 samples have been collected. There's been different ways of technology that have been applied. I should comment, the short-read sequencing, just because it got started earlier, is ahead. There's more than 500,000 samples now that have been sequenced using short-read sequencing. But I'm very excited to announce, the long-read sequencing is also starting to come on board in really a massive scale. So to get started, a couple years ago, we launched a pilot study. And this was at small scale, just looking at a few cell lines, but kind of the headline from this was undisputed superior performance, looking at things like structural variations, tandem repeats, segmental duplications, and other complex variation. Even in kind of sort of single nucleotide variation analysis, thanks to the long-read sequencing, you get better mappability. So you can kind of pierce into some of these repetitive elements and just get better resolution of SNPs and small indels. So for this talk, I'm really focused on structural variation, but even if it's really single nucleotide variants or kind of more simple types of variation that you're interested in, I would argue that long-read sequencing is really superior there. And crucially, it's not just in the centromeres, it's not just in these like really complicated parts of the genome. Even in genes that are known to have medical relevance, we know that we've been able to measure now that you get superior mappability, superior variant recall in those clinically relevant genes. And then finally, we can get improved phasing as well, right? Again, phasing is all about figuring out which variants are on which haplotype, aka are on which molecule. If you have long single molecule sequencing, of course you get better phasing. because you can just sort of get a direct readout from the molecules directly as to where those variants fall. So there's a pilot study looking at cell lines that sort of concluded that yes, long-read sequencing was superior, and based on the accuracy of the variants, and then also very practical considerations about the throughput and the cost of the platforms, we selected HiFi sequencing to be our workhorse for the All of Us program. And then so moving forward here, the story I'm really going to tell you is about our so-called phase 1 effort, where we have sequenced 1,027 All of Us participants who are self-identified as Black or African American through the All of Us program here. And all of those were sequenced using HiFi sequencing. And I'll tell you a little bit about their genomics data, talk a little bit about some of the variation in, say, genes that are known to be clinically relevant. And then in the end here, I'm going to tell you about our initial study with what's called a phenome-wide association study, where because we have access now to their sequencing data, as well as to their medical records, we're looking over hundreds and hundreds of conditions and traits to be able to do an association, asking, what types of variation, if any, are associated with all these different traits that are present there. And I think I have some really exciting data to share with all of you that kind of really indicates the importance of structural variation across many, many different traits here. So the first part is sort of the sample selection and the genomic data that were generated. Again, we wanted to sort of maximize our ability to capture genetic variation. Many studies now going back to HapMap and 1,000 Genomes have indicated people of African descent, just because of sort of human population demographics, those individuals capture the most structural variation and also single nucleotide variation and other variation very broadly. So from this, based on sort of the practical considerations, we identified just over 1,000 individuals that we are using for this phase 1 study that we selected for HiFi sequencing. Shown here is a PCA plot of the people in these little diamonds that were selected for the study, and we're overlapping this with other sort of cohorts like 1,000 genomes to put them into sort of genetic context. These were people that were self-identified as African-American individuals, and indeed, the vast majority of these individuals fall along PC1, which canonically captures individuals with African continental ancestry. There are a few people off diagonal, and what those people really indicate is some sort of admixture between their African composition as well as other kind of continental regions around the United States. These by design, it didn't come from just one sort of geographic region. By design, we try to select people geographically distributed around the United States so we can really have a comprehensive resource of all their sort of genetic background, and also kind of access to different hospital systems, different EHR data that would be available there. So across all these different individuals, we could just sort of tally up some of the sort of medical records that are available using kind of standard ontologies, using something called OMOP and the Common Data Model, CDM. Depending on the individual, we had more than 1,000 sort of medical records that are associated with them, in terms of different procedures that are going on, different observations, measurements, exposures, other sort of conditions that were present there. Now, this was designed to be a cross-section of Americans and a cross-section of their health. So you see, as you would expect, a cross-section of conditions that were present there. Common things like metabolic processing, endocrine, cardiovascular indications, GI indications were quite common. 40 to 50% of the participants, we had EHR data on those sorts of conditions, and then a long tail, right? From a variety of neurological, mental, different infections, pregnancy conditions, and beyond. So it's really designed to be a cross-section of Americans and their health. And that's sort of, in your mind, that should be the mental model of the types of the conditions that are present here. Now, for very rare conditions, right, because this is a cross-section, we're not going to have a particular enrichment for rare conditions, although by, happenstance, we may have a few cases of different rare conditions, but in general, we're going to be focusing on those common conditions because of this cross-section. We're going to be really focused on that. I look forward to kind of future studies where we can follow up in great detail on some of these rare conditions, and you'll see examples about how that might be impacted through this study on some of the data I'm going to present here in a second. So this is a summary of the data and the variant calling that was performed here. So we had about 1,000 people. For practical considerations, when we launched the project, to keep it sort of practically affordable, we were targeting sort of modest sequencing depth here. So this is 8x coverage. I should comment, we did a lot of analysis, both theoretically and also empirically, to ask the question, how successful are we at recall and specificity at 8x coverage. Of course, more would be better coverage, although when we're looking at thousands of samples and then beyond, into tens of thousands of samples and beyond, there is this sort of practical reality. I would say this all predates the SPRQ-Nx chemistry. If we had to launch this initiative today, we might sort of dial up that dial in terms of the amount of coverage. But at the time that we launched this, we were sort of picking this modest coverage. Although we did a ton of benchmarking, we did a ton of analysis. This gives us remarkably good sensitivity and specificity at 8x coverage. There's some very old theory that goes back to the original Human Genome Project, the so-called Lander-Waterman statistics, that says at 8x coverage, we're going to get almost more than 99%, 99.9% of the genome resolved at 8x coverage. And then because of that, we get excellent sort of variant calling as a result, or at a per person basis. And then using latest and greatest machine learning technologies and also latest and greatest merging technologies, we can get sort of pristine population cohort variant calling through these data. So there's a lenient sort of call set that's about 1.2 million variants over the population that is really sort of prioritizing sensitivity, and then a really, really strict call set of about 600,000 variants that is really optimized for specificity. In this study, we're a little bit conservative. We really wanted specificity. So most of the analysis I'm going to present next is looking at this cohort of about 600,000 variants across the population. Although on a per person basis, we're talking about 25,000 variants or so. I really want to also on this slide credit the amazing team that went into this. At the end here, I'll give you a pointer to the preprint that describing the study that has more than 100 authors involved. Here, I'm just calling out a few of the key people involved in the structural variant analysis. This was, great partnership of many people. Here, I'm just calling out a few: Kieran, my postdoc Iris Quihi, Sam, Steve, Fabio, and many more. There's just kind of too many people to go through all of them, but it was really a team effort with major players at Hopkins, at the Broad, at Baylor College of Medicine, at University of Washington, at Miami, and beyond. So this is really a sort of a nationwide effort here to bring together sort of leading experts to really push forward on this analysis. And it's really been a pleasure working with everyone. So another thing that we can do, and this will become important later in the talk here. So most of the analysis is based on HiFi sequencing, right? This is going to let us sort of access parts of the genome and types of variation that have never been seen before. And in sort of round numbers, using HiFi sequencing, using long-read sequencing, for structural variants, you get about 2 to 3 times more variants are detectable using long-read sequencing. Again, as I said, about 25,000 variants per person using, say, long-read sequencing. Even through T to T assemblies, about 25,000 variants. Compared to with short-read sequencing, even using state-of-the-art pipelines like GATK-SV or Parliament2, if you're just using short-read sequencing for structural variant discovery, you may capture, 8,000, 10,000 variants. Things like large deletions are pretty easy to capture using short-read sequencing. Large duplications are pretty easy to capture. But things like smaller deletions and then specifically insertions are very difficult to capture using short-read sequencing, right? If the insertion is larger than the read, you'd have to do something like de novo assembly and then try to map them. My lab and other labs have worked on this problem. It's just a hard problem, right? Short-read sequencing, it's hard to assemble. Insertions tend to have some repetitive elements inside of them or flanking them. It's hard to resolve. But if you know that there are deletions, if you know that there are insertions in the population, there are strategies that you can use called genotyping and imputation. If you know about where these variations exist in the population, and you can ask a much more focused question. Here's short read data. Ask this very focused question. Yes or no, there's this variant that I'm interested in, yes or no, does it exist in this person? Is it heterozygous? Is it homozygous? There's actually some mature technologies available to be able to answer that question. In fact, PacBio and others have worked on this. For all of us, we've adopted this pipeline that uses something called KAGE that looks at k-mers that are present in the structural variant, right? So at a deletion, you'll get a junction where the two parts deleted come together. In an insertion, you'll get k-mers, distinguishing k-mers right at the junction sequence where you branch out into the insertion. And then inside of the insertion as well, you'll get k-mers that can uniquely identify the presence of that insertion. So that would be our genotyping strategy, where we can revisit short read sequencing and then ask if this insertion or this deletion occurs. And then using kind of evidence from the genotyping as well as flanking SNPs, as well as flanking indels, you can do imputation to further refine this sort of process of asking, is this structural variant present in the short read data set? So our study begins with long read sequencing, begins with HiFi sequencing, but then using this genotyping and imputation, we can visit other data sets with short read data to get a look at sort of the variation that's present there. Benchmarking using long read using data that's matched with long read sequencing and short read sequencing from say the Human Pangenome Reference Consortium, our accuracy is better than 90%. Although I will comment outside of tandem repeats. Inside of tandem repeats, where the short read data is really confusing, you're going to be really challenged to get accurate genotyping and imputation there. But outside of that, we get excellent quality. So we can discover variants in all of us, and then we can visit, in this example, 1,000 genomes data, so that we can comment, even though these variants are from people of African American descent, we can notice that many of the variants are common in admixed American populations and European populations, East Asian populations, and South Asian populations as well. So it's really a nice resource globally, even though we started with individuals of African American descent. And then also this pipeline will be very valuable later, so we can tap into additional data sets. So again, we can sort of characterize where these variants occur. We can characterize that many of them do occur in genes. We can characterize many of these variants do occur in regulatory sequences. And we can kind of comment many of the variants are shared across all populations, although some of them are specific to African Americans in both the gene variations and regulatory regulations. And then there's sort of a long tail that there is enrichment across other parts of the world as well. Okay, so we get this really nice data set from the HiFi data. It's of primarily self-identified African Americans, although it does let us comment globally about the role of structural variations in a sort of a global scale. So a natural question is now we have all these variants, we can kind of look through them. A natural question is like, do these variants intersect interesting genes? That's sort of a natural question. Now, on a per person basis, there's great sort of selection against structural variants falling inside of genes commonly. But because we have 1,000 individuals here, we get a nice look, at a population scale. And it's actually quite common for structural variants to intersect gene bodies and also within exons when we're looking at this scale, especially when we're looking at, these are, kind of common demographics. Many people have cardiovascular disease. Many people have other conditions here. So in this biobank setting, we see lots of variants intersecting genes and exons. So here's sort of a tally of how often, pick your favorite set of genes. This is OMEM. Here are genes associated with inherited diseases. Here's genes associated with cancers. Here's sort of important ACME sort of identified genes. And yes, structural variants are enriched in, segmental duplications. They're enriched in centromeres. They're enriched, in sort of more complicated parts of the genome, but they are also occurring in all of these medically relevant genes as well. And that's really important to know as we kind of zoom in here. Here's an example. Again, even within the gene exons, we're finding many examples. Down here at the bottom here, here's an example of an SV inside of a gene that falls in the MHC complex. and is sort of a key gene associated with immune-related conditions here. So it's really, really important to be able to study these as we're kind of getting a detailed profile of patients and all the variants that are present there. Another thing we can do is sort of scan through this collection. There's a really nice tool called CADD-SV that makes a prediction about how pathogenic a given variant is, based on a variety of different features associated with it. So, is it an encoding sequence? Is it a known regulatory sequence? Is it intergenic? What is the conservation of all this sequence? Other sort of properties of the variant there. This is CADD-SV. I was not involved in the development of this tool, but it's really nice here. The score that you get is in a sort of a Phred scale score, where a larger number is more and more confident that, yes, this is very, very likely to be pathogenic. Similar to kind of a quality value with sequencing data, quality value of 20 is like pretty confident. As we get into 30, 40, 50 and beyond, these are like almost certainly extremely pathogenic variants. So as I was mentioning, right, there's great through evolutionary selective forces, pathogenic variants are going to be rare in a population, especially a population that is, not sort of focused on a disease cohort, but is just sort of a cross section. So most of the structural variants that we see have sort of modest, scores of below 10. But then there is this subset of variants that are, very, very likely to be pathogenic. And then most of these, more pathogenic ones are only detected through this All of Us study, where we're looking at the scale of 1,000 people that are present here. So there's on the order of almost 300 variants that are very likely to be pathogenic based on CADD-SV. 170 of these are going to be overlapping medically relevant genes. Most of these have never been seen before, right? This is the importance of doing long-read sequencing in your own cohorts, in the power of that HiFi sequencing to detect structural variants that have never been seen before, including variants that are very, very likely to be structural variant. Now, the thing about CADD-SV, it's just a prediction. We like it. It gives us, a really strong indication. But then as we dive into some of these examples that have, really, really strong scores, we see really interesting patterns here. So on the left, here's an example of an insertion. That's what this little I here in IGV is giving you. This is an 52 base pair insertion. It's in this gene, which is in a glucose transporter. And it turns out, once we sort of identified this, CADD-SV guided us to this variant. We didn't know this going in. We looked at sort of the EHR data for this individual, and the individual did have type 2 diabetes, and which was highly related to this metabolic impact, which is very likely to be associated with this glucose transporter. Here's another example of a 536 base pair insertion, a very strong CADD-SV score. Again, we were sort of guided to it through the SV calling, guided to it by CADD-SV. And then we noticed it was in a gene that was a known regulator of asthma. And what do you know? The individual having it did have asthma, in fact. We call this a little bit phenotype gazing. It is not necessarily conclusive here. Of course, we'd want to have follow-up studies to look at this. These are very rare variants. In fact, both of these variants only occurred in one person. But now that we know about them, we would, what was ongoing now is look at other sort of diabetes cohorts, look at other asthma cohorts to see if we can see evidence for these variants, more commonly. We're really excited about this as a discovery tool to look at candidate variants, candidate genes, and then go be able to follow up on them with these with these variants that are predicted to be extremely pathogenic. Another thing that we can do is sort of mine, kind of regions that are known to be sort of carrying important clinically relevant variants. So what we did here is we went to the GWAS catalog, right? This is a very famous catalog that sort of integrates, all of the known variants that are associated with different traits and different diseases through genome-wide association studies. There's on the order of 1 million SNPs now that have been sort of labeled as being at least statistically associated with different traits and different diseases here. So we can look at all those SNPs, and then we can just ask a very simple question. How often is it the case that there's a SNP that is nearby? In fact, how often is the case that there's a SNP that is in strong linkage disequilibrium with one of the structural variants that we were just able to find through this All of Us study? And it turns out it's quite common. We have, depending on sort of what sort of cutoff you're interested in for linkage disequilibrium, there's hundreds to thousands of structural variants that are nearby and in strong linkage equilibrium. And kind of our hypothesis is, sometimes the SNPs that were identified are likely to be, the causal ones. But, just through the statistics of how GWAS works, those lead SNPs are often just markers for underlying genetic lesions. And our kind of a hypothesis, our kind of our goal here is to be able to think through, well, maybe these SNPs that were reported through prior studies are really just tagging structural variants that are actually doing some of the work here. So this is our way to kind of get a survey of how likely that is, a potential feature here. And the headline being, it's quite common across many, many different traits, hundreds of traits, thousands of diseases or disorders. We're finding a structural variant in very strong LD with those GWAS SNPs. And those are very interesting for kind of follow-up. Here's a couple of examples, again, where the structural variants were discovered in our All of Us study. They were kind of in the high linkage disequilibrium with SNPs that have been, kind of reported in the literature. On the left here, again, here's one associated with asthma. Has a nice p-value through GWAS 10 to the minus 10th. Here's a structure variant, again, another insertion that you can really only find through long-read sequencing. And again, it has a really, really strong odds ratio. It's a really, really strong predictor of having asthma. Previous GWAS studies, didn't have access to the structural variant. But in fact, some of the metrics here report that this is a better indicator for asthma than the SNP that is reported in the literature. On the right here, another example. Again, the GWAS catalog has this nice SNP, 10 to the minus 10th, has an odd ratio of 1.19 associated with breast cancers. In very high LD with the structural variant that we're able to detect through All of Us, it has an even stronger odds ratio. So we think here, again, here's another example of a deletion that is readily detected through long-read sequencing, through HiFi sequencing, that has a stronger odds ratio, really nice p-value. So here's a really exciting result as a potential risk factor that was just detected using the long-read sequencing in combination with with known GWAS SNPs. So again, there's sort of growing evidence that that these structural variants are playing an important role. It could very well be the case that many of the SNPs that have been reported in the literature are actually just, effectively tagging these structural variations that are likely to be doing the work, especially through insertions and deletions of these larger elements that can carry, important regulatory elements beyond, what a single SNP can be reported. Famously, most of the most significant SNPs that are reported through GWAS are in intergenic space. It's been kind of confusing as to what they're doing. My personal belief is that often those SNPs are tagging structural variants that are disrupting regulatory sequences. Okay, and then the last part of the talk in the last 5 or 6 minutes here is to go through our work, doing what is called a phenome-wide association study, a PheWAS study, where we can do a straight up statistical analysis association of the structural variations with all of the medical conditions of the All of Us participants. So here we're going to use both the structural variants directly reported out of the long read data, but we're also going to tap into that genotyping imputation panel that I mentioned earlier. And the idea here is because we're doing an association study and our cohort has a cross-section of different traits and diseases, we can use that imputation and genotyping to kind of expand our population size from on the order of 1,000 samples up to the order of 10,000 samples. So here's a way where we can kind of really boost up the numbers, which will increase our power for detecting these associations, boost our power of just having really conclusive results here beyond some of the results I just showed you using just 1,000 samples that we have here. So what is shown here is, again, we start with variants detected in the long read data. We go use KAGE and GLIMPS to go ask the question, yes or no, are these variants found in the short read data through 10,000 other participants? We worked really hard on this to kind of clean up some of the noise here, but now we get really a nice match, say, allele frequency distribution on the SNPs, on the SVs. We have great confidence in the structural variants in particular that we can find in this way. And then through this data, now that we have access to 10,000 samples, we can just sort of ask which sorts of conditions are common in this population. And there's close to 500 different disease phenotypes, ranging from things like immune disorders, cardiovascular disorders, musculoskeletal disorders, and beyond. So across many different common conditions here, we have many different phenotypes now matched with this excellent genetic information associated with them. So now we can go through here, and then we can just sort of visit every structural variant, and we can visit all 492 of these different medical conditions and just ask, is there an association between the structural variant and the condition? The headline being is most of the time, yes, there is a statistically a significant association between one of the structural variants and the traits of interest. That's exciting. And then furthermore, we can ask sort of a related question. Now we have great structural variant data. We also have great SNP data. We can ask sort of a follow-up question. Okay, yes, we find an association to the structural variant. We can also ask, is there an association between any SNPs that are nearby, right? Variants don't travel independently. They travel in haplotypes. Structural variants are going to be in the background of many SNPs there. So we can ask the question, what is more significant? When we find that there is an association to a structural variant, what is more significant, the structural variants or the SNPs that are nearby? And that's what this sort of complicated plot here is trying to show here. So on the x-axis is the significance of SNPs. On the y-axis is the significance of structural variants. A lot of the variants indeed are along this diagonal where it's sort of a toss-up. Both structural variants and SNPs are playing an important role here. But a key takeaway for the audience here is, in fact, most of the time, most variants are above the diagonal, which means that most of the times the structural variants, at least statistically, have a more significant role in the disorder than any of the SNPs that are nearby. This is a really important finding in my mind, whereas most of the time GWAS has been focusing on SNPs, and again, that's been driven by the technology, starting with microarrays, and more recently short-read sequencing. Most of the time, it's actually the structural variants that are more significant. We just didn't have the right technology. We didn't have HiFi technology until right now. But most of the time, it's the structural variants that are more significant. This is an incredibly important conclusion for clinical genomics. Most of the time, you should be looking at structural variants beyond the SNPs. That's where those really important variants are going to live. That's really, really important here. Furthermore, as you kind of partition the space about the significance of those variants, here measured through relative risk, as a function of the allele frequency. Again, there's great forces that keep, especially pathogenic variants are going to be relatively rare in the population. Most of the really important variants that are present there are going to be pretty rare. They're going to be at the level of 1% or fewer. That speaks to how important it is to be doing population scale sequencing, right? If you have a small cohort, of 100 people, you're just not going to see variants that occur, 1 in 1,000 times. You're just not going to see them very often. Unless you're doing, pedigree type analysis, you're not going to be powered to be able to really comment on their significance. But by scaling up to 1,000, 10,000, 100,000 or more people, that's where you're powered to be able to see these really, really important variants that have, odds ratio of, you're 25 times more likely to get a certain condition. That's a really important clinical finding that you're able to get through the power of population scale sequencing, especially HiFi sequencing for all these structural variants. Here's an example looking at a heart condition, hypertensive heart failure. This is a Manhattan plot mapping out the p-value of all the variants kind of around this gene. It's a classic sort of Manhattan plot where you get that sort of nice distribution where, in fact, in this example, the lead variant is one of our structural variants that was discovered through HiFi sequencing. Kind of unsurprisingly, other variants with high p-values are other SNPs, and indeed, they are in high LD. So again, when I think about the GWAS catalog, I think it's often like this, where they've been looking at SNPs and they were just blind to these structural variants that were out there that are actually more significant, are more impactful. They lead to just higher odds ratio. A couple important things. So this particular variant is a deletion. When we look at how it impacts the gene that is present there, it really truncates the gene, leads to really a highly disrupted protein product that is there. We would comment that this gene is known to be associated with cardiomyocyte shape and development. So it completely makes sense why this would be related to heart failure. We're currently in the process of validating this finding in other cohorts, but already the results there look really, really promising. Another kind of question that we can do is many of those variants are intergenic, but we can also follow up and look specifically at coding variants. Same sort of scenario. This is that same sort of plot where we can measure what is the significance of coding variants that are structural variants relative to the background of all the SNPs there. Again, most of the time it's the structural variants that are more significant. Again, it's going to be these relatively rare variants at the level of 1 in 1,000 that are going to have our strongest odds ratio that are going to be really important. Here's an example of one of them. Again, this is a collapsed lung disorder where the odds ratio is incredibly important. The significance of the structural variant is much greater than any of the SNPs that are nearby. And then importantly, here's an example where the structural variant is not in particularly high LD with any of the SNPs that are nearby. And the reason for that is this happens to be in a fairly repetitive region that can only be accessed through long-read sequencing. Repetitive regions have different mutational profiles than the surrounding SNPs. So this is, it's present, it's really, really robust in the long-read data, and you're invisible to this using short-read sequencing. It introduces a stop codon, again, highly disruptive to the protein. In mice models and other results, we see abnormalities associated with collapsed lung. So it kind of completely fits why this would be a gene, why this is the type of variant that would be really important. Again, can only be discovered through long-read sequencing. In particular, HiFi sequencing gives you this pristine readout about the structural variant that is at play here. So just to sum up here, structural variants are major, major drivers of traits, of clinical conditions. Most of the time, about 2/3 of the time, in particular, say insertions are going to be missed through short-read sequencing. You just have to do the long-read sequencing. They're incredibly important. Most of the variants that we're finding that are associated with these different traits are missing from short-read call sets. Most of the time, 2/3 of the time, it's the structural variants are more significant than any of the SNPs there. It's really, really important that you're looking at these as you're doing any of your clinical cohorts here. They're major sources of disease-relevant risk, contributing to the missing heritability of complex traits and diseases. Also, I would comment, also critically important for rare disease as well. That's been reported through my group and other groups around the world. Just really incredibly important across the board there. If you're interested, I encourage you to check out our preprint. It's under review right now. We got very positive first reviews. We're following up on some of the questions that are there. Look forward to a revision in the coming months here, kind of looking at even just strengthening the results that we've had so far. Again, I'd like to really thank the entire team here. It's been an awesome team effort diving into this. In fact, in the paper, there's so many juicy results here about tandem repeats, about different haplotype structures, EQTLs, some of the pleiotropic effects. I really encourage you to check out the preprint just from the interest of time. I couldn't possibly go through that in great detail. And as a little teaser here, this is just the very beginning. So this is our first 1,000 samples in phase 1. We're just finishing up the second phase of 10,000 samples with HiFi sequencing. I look forward to presenting these results at ASHG coming up later this year to get a first look at this much larger cohort, how this has tremendous impact, very, very broadly in disease in very different traits. Again, huge thanks to the huge team of awesome, awesome, awesome people working together. Also, within my own group, I'd like to thank my lab members, especially my postdoc, Iris. She's just been truly spectacular to work with. It's also been just spectacular to work with so many great collaborators in the world. Genomics is all about team science, where it comes together through the integration of ideas. Also, I'd really like to thank PacBio for the tremendous support and also technological advancements that has made a dream come true to be able to look at the foundation of all these different conditions. So thank you very much for your time and attention. Please follow up with us if you have any questions. It's just a delight to talk about all of this. Thank you, everybody.
Thank you for attending this webinar. Unfortunately, we don't have time for any live questions, but we will get back to you offline to address any Q&A. We hope you enjoyed today's presentation. Please visit us at pacb.com to learn more. And again, we'd be grateful if you could take some time to fill out the short post-webinar survey. Thank you.